All right, so this is the final presentation on the function of the Bradi5g15190 gene, and we're going to talk about some of the experiments that we did and what we learned from them. So the first thing we're looking at here is the phylogeny of the gene, and we found that it has many relatives, some of them more close than others, but it has relatives in several different organisms, which suggests that it's possibly conserved over multiple organisms. And its closest relative was found in Oryza sativa japonica, or rice plants. So we used that later in looking at the literature, which we're going to talk about now. So this is the gene homepage for the gene, and it shows the name of the protein, which is protein trichome birefringence-like 2, and it also shows the two known domains of the protein, which are the PMR5 domain and the acetylesterase domain, right? So we couldn't really find virtually any info on the acetylesterase domain. We could find it in some other organisms, but not really in plants. So we didn't have any information on that, but we were able to find some stuff about the PMR5 domain. And basically what we found is that it's a domain that's been long known in Arabidopsis to confer resistance to powdery mildew, right? That's the PMR. And what the paper that we found showed is that the way it does that is by adding acetyl groups to cell wall pectins, right? So they measured the activity of the domain in the presence of certain pectins, and they found that the activity went up and that there were more acetyl groups on those pectins. So they hypothesized basically that it functions by transferring acetyl groups to those cell wall pectins, which has a big effect on the response to stress. And we found a similar thing on the TBL2 protein in Oryza sativa plants, right? And from the phylogeny, we knew that that was the closest relative of our protein. So what we found here was basically that in knockdown experiments, that there was a lower level of acetic acid content in them, right? So when they had mutants where the TBL2 gene, and they also studied the TBL1 gene, but we were more focused on the TBL2 because that was the analog of ours. What they found was that there was lower acetic acid content, and they knew that that was a marker of decreased xylan acetylation, right? And xylan is another cell wall polymer. It's a hemicellulose. And so this is kind of similar functions here, right? So basically what we're able to glean from this is that it seems pretty likely that the function of our gene is to transfer acetyl groups to certain cell wall polymers. And that has a huge effect on their response to biotic and abiotic stress, right? In this study, they found that the knockdown mutants were more susceptible to blight disease. And so it's really important, but they don't really know too much about the function of why acetylation in the cell wall is super important, but they know that it is, and that's basically our best idea of how our gene functions. And so Vanessa's going to talk a little bit about the mutant stock that we had and kind of the information we weren't able to get from that, and she's going to talk a little bit more about expression.
After we extracted the DNA from mutant stocks, we ran PCR to amplify the mutated region of the genome, and we couldn't get DNA bands to show up on either trials of our PCR. We only got bands at 100 base pairs, which are primer dimers, and our DNA should be around 700 base pairs. And since we couldn't get DNA bands to extract to send for sequencing, we don't have sequencing results to analyze the genotype of the mutant stocks. Then we found that our gene is mostly expressed in roots. From the EFP data on the right, we see that it's red in roots and yellow everywhere else in the plant. That means there's high expression of our gene in roots and low expression everywhere else in the plant. We see a similar thing in the graphs to the left, high expression in roots and low expression in other parts of the plant. We also found that our gene is regulated by a thermocycle, which means that it is affected by hot and cold conditions. For our experiment, we wanted to test the effect of IAA on our gene expression because we know that IAA is supposed to stimulate root growth and that our gene has high expression in roots. We hypothesized that with IAA, there would be greater expression of our gene in the roots. And so to set up our experiment, we grew 40 brachypodium seeds on 4 agar plates for 7 days, transferred 20 seedlings to an untreated plate for control, and 20 seedlings to a plate with 10 micromolar of IAA. We collected the roots after 3 hours to extract RNA and analyze the root material with qPCR. So from the RNA extraction, we can see from the gel that there are bands at 1400 base pairs and 800 base pairs. Those are believed to be rRNA, which are the 25S and 18S subunits. Then from the qPCR, we use the CT values of all the samples to analyze the results, and found for our experiment that there's no significant difference between our gene expression and the housekeeping gene of the untreated and treated samples. The p-value was greater than the cutoff of 0.05, with it being 0.087. So there's no significant difference. And for the given cDNA data on the right, we could not determine the p-value because there was not enough mutant data samples.
All right, so just in conclusion, what we know, or what we can say with decent confidence about our gene, is that based on the domains it contains and based on its most closely related genes and proteins, is that it's likely involved in the acetylation of certain cell wall polymers. We can't say with much certainty what polymers those are, but it's likely involved in acetylation of cell wall polymers, and in turn, that influences the response of the plant to biotic and abiotic stress. And we know for certain that it is almost exclusively expressed in the roots. And it also seems fairly likely that its expression is regulated by a thermocycle. So our best guess of the gene is that it's a protein that functions in acetylating certain cell wall polymers, but must be mostly in the roots. So it functions in strengthening those cell walls in the roots and the growth of the roots. What we don't know is really anything about the mutant stocks that we were provided because we didn't have any effective PCR, and we can't really say with confidence what the effect of IAA on the expression of our gene is. But yeah, so that's really what we know about our gene. At the end of the day, I guess it really isn't that much, but it was only one semester of study. And thank you for watching and listening.